All right. I just have one slide listing questions because I think we've all had a very great set of talks already this morning. So I think in the context of this discussion about variant discovery and association, I think the questions that I think we're all challenged with is figuring out what is the right role for NHGRI. What are the scientific issues? What are the important questions to ask, not just in the context of all of science and understanding the genome, which is probably beyond what NHGRI on their own can do, but what are things where we can make a big difference and that maybe wouldn't be done as well or in as comprehensive a manner without the focused investment here. In the context of the conversation we just had in this last little bit, I challenge you to think about what your advice is in terms of what sort of samples those questions translate into, whether it's case controls, cohort studies, biobanks, families, diverse populations, unique populations, phenotyping, what's the right level of phenotyping that we want, what's the balance between collecting these phenotypes up front and having the ability to recontact and do experiments later, what are the right sets of genomic data that we should be generating, if any, right? The technologies we should be promoting. Function, I think a few times we've heard about ENCODE and other projects that really create a lot of value for the community. We also heard several times about data access and interoperability. And I think like Jonathan, I think it's important to think about, how do we keep a vibrant community? And I think this is about data sets and so on, but it's also about getting new people to come and join and bring their ideas. And it's also about having tools that facilitate some of these things that we imagine. If we think it's really important to be able to combine data across cohorts and studies, let's come up with ideas for tools that make it really straightforward to do and to do that in creative and new ways. So I'll just stop there and open it up. I don't know if my panelists want to chime in with any thoughts or just open it straight to the room.
Happy to have questions.
Yeah, I'll make a comment. Is this on? So as I see the field moving forward, we are going to have increasing disparities in healthcare. And why are we going to have that? Because we have a tremendous amount of information on populations of European descent. We really don't have a whole lot on other populations, in particular African ancestry populations. So as this technology and as these things come forward, just the practicality is that it's going to increase disparities. And it's also going to increase disparities probably in rural versus urban areas, with the urban areas having access to this technology. And as we design all of our studies, we need to be thinking about how do we design to reduce disparities, because it's happened now and it's just coming. And it's I'm not trying to blame anyone, but it's just a fact, and we should hit this head on.
No, there definitely is a big balance between, at the cutting edge, there's things that we try or are tried in the US versus things that are accessible to the rest of the world and to everybody. And you need to spend effort on making things that are working accessible in addition to developing the greatest new thing.
And I'm talking about disparities within the United States.
Yeah. Cool. All right. I'll open it up to the room. Kind of surprising this. Oh.
Hey, I think one of the things that is really, really important to tackle in the next 10 years in this space is the governance issues associated with electronic health records and integrating them with this kind of data. The power that I think the community would get from some sort of solution to this problem of having really well integrated medical records and genetic records would be enormous. But I also think that some of the right people need to be at the table in terms of large healthcare organizations, insurers, et cetera.
Yeah, just to follow up on one thing that Jonathan was touching on is I spent almost three 3 decades as sometimes a lone voice to try to convince that there was value in the genome and the data from the genome towards druggability. And so with regard to a biotech and pharma, which is my circle, and we saw the ascent and the decline of F2. We saw the rise and fall of GWAS as we had spectacular failures of billions of dollars invested in a lot of targets that made a lot of sense. Base 1 being one of the most impactful crater. And so with regard to understanding the functional genome, I really resonate with that. And how do we take the combined efforts here and really find tools that help us to refine what we consider as a druggable target? And that doesn't mean as oral once a day. I mean, that means as a therapy of any sort. And so be able to be able to sort the wheat from the chaff would give us a lot better impact from our combined efforts, from the monies expected, and the tools to help to understand that. So is there anybody here from the Druggable Genome Initiative or any of those aspects? I'm not sure who's here from that. Or Jonathan, do you want to comment on that? Because the applicability towards the amount of money and attention being invested in biotech and biopharma is incredible. It's at a key point in time, and to make our efforts here impactful to that is paramount for the next decade.
I think you're absolutely right about that, and I'll make a couple of comments. Certainly, within Alzheimer's disease and most neurodegenerative diseases, the focus initially, because it was what the FDA would allow us to do, was to look at people who were already affected and see whether or not we could slow or stop the progression, things like that. And almost assuredly, that's too late in the process. And we really should be doing these, trials 30 years before people 20, 30 years before people get affected, because we think the process starts actually way back then. Then you get into, issues about, is that appropriate to put somebody in a clinical trial for something that's not going to happen for 20 or 30 years? And that's another whole, that's an ELSI issue that really should be discussed. There's also a second aspect, which is, I think we can be smarter about identifying the targets, right? So one of the things that has happened up to, recently, is we find a gene, we find a variant in the gene, and off we go. When what we probably should be looking is what's the impact on the protein, what's the impact on the structure of the protein. For example, taking the variants that we see and projecting them into the 3D structure of the protein to find out whether it actually makes a difference or not, or whether there's a key part of the protein that we really want to look at or should be investigating in more detail. So, and those things are just starting to happen now.
Hey, and please identify yourselves before stating your comment.
There you go. Oh, it's on. All right. Thank you. I'm Katie Pollard from Gladstone Institutes in UCSF and Chan Zuckerberg Biohub. I have a question either for the panelists or people in the audience. Earlier, we heard about the hard parts of the reference genome, like repetitive elements and structural variants. And I'm wondering, in this context of variant discovery and association, what the cutting edge is for studying those, not in the reference genome, but in large numbers of people.
Well, that's a hard one. I will say one of the things, just to take a tiny slice of that, one of the things that we've been struggling with, again, in the Alzheimer's disease sequencing project, is trying to identify the structural variants using the short read data. This is, it's okay when we're looking at really small indels and things like that, but when you start trying to build across the across the short reads, this becomes a real problem. We've tried, I don't know how many different programs, out there that tried to rebuild things, and it turns out that some of those programs are pretty good for some sizes, not other sizes, and how do we put that all together? It's been a real struggle, and we've actually spent like 3 years, and I think we now have an initial version of deletion deletion variants that we may be able to start to look at. It's a huge problem, and I really think that, my personal view is that that's going to come from probably long read or things like that. I mean, 10x genomics helps some, but it's not perfect. There are various ways of doing
So
that.
if I was going to chime in there, I think
Yeah.
the current state of the art in terms of examples of structural variants that are being implicated in common diseases, usually, with whole genome sequencing, someone focusing on a particular locus, developing algorithms specifically to dissect variation in that locus and spending time digesting it, whether it's C4 or whether it's factor mutations and things like that. Usually those initial analyses are in relatively small sample sizes for now, and then to look at them in very large sample sets, I think at the moment we rely on arrays and imputation once they've been characterized in detail. Evan has got probably a different take and is...
No, actually, well, I think what you said is absolutely right. I just want to make a point, because maybe it wasn't made strong enough before, is that long-read technologies are really increasing by threefold our ability to detect this variation. So it's tremendous. And it's like a whole new form of human genetic variation is being uncovered by it. A couple features of it that has been actually documented by several groups already has been that these variants hit above their weight in terms of genome-wide association, intersection, as well as in terms of EQTLs. Not a surprise. You delete or duplicate a big swath of sequence, you're going to have a larger effect. The bottom line is that by simply doing Illumina whole genome shotgun sequencing, you are detecting probably on the order of only 25% of the structural variation, even if you run 12 different collars and you take the most sensitive set. Having said that, the solution is long reads, but it's interesting when you look at the long read sequence and assembled genomes, the regions that they don't assemble are some of the most copy number polymorphic regions in our genome. So 50% of the gaps in a genome that are now assembled are mapping to the largest, most structurally variant copy number areas of our genetic code. So 50% of all the structural variation in the genome is mapping to the gaps. And these are the regions that contain the genes, and these are the areas that are going to have high frequency of recurrence, which makes them, in many cases, unimputable. So you actually have to genotype directly the variant. So this is a big thing, and I'm sure we'll discuss it much more in the breakout session. But I think this is something that people should think about when they sequence a genome deeply and they check the box that they've run 3 collars and they've got a structural variation call set and they've done imputation, is that there's still large swaths of gene-rich territory that are not imputable because they're not there.
Nothing is not imputable, I think, but...
So coming back to the kind of challenges in the Alzheimer's field that actually probably are just a canary in the coal mine for every other field. Your slide where you showed all of the variants that have been identified by sequencing, for example, listed SELF1 as one of those genes. But we figured out later on that SELF1 is actually not the gene that's the causal variant there. It's actually PU.1, which once again ties back to the microglia. But what I'm trying to get at is a lot of these variants are falling into very complicated regions where actually knowing which gene is the one that's being affected by this variant is very difficult. And at least my experience from watching the Alzheimer's field is that people see all the variation that's falling in the putative regulatory elements, and they want to focus on the rare variants that are falling within the coding sequence of genes because they know what to do with that. And so they go do that. And then they try to throw in single-cell RNA-seq of the particular brain regions because they're hoping that that's also going to somehow give them help interpreting the data. They get the data, and they still don't know what to do with it. So the bigger thing, I think, is for NHGRI maybe to figure out how to assist all of these other projects in trying to come up with ways of looking at this data, analyzing this data, putting things together so that the institutes that have specific focus on specific diseases then can leverage everything that NHGRI could come up with. I don't know how to come up with the perfect disease for HDRI to sort of test drive this in, but there's a place for us to develop the tools that all of the other consortiums and all the other efforts could then use in order to be able to leverage all this data they're getting, but not being necessarily able to interpret at the first or second or third try.
Thank you. Sharon.
Yeah, I wanted to bring up 2 points, you know, and I'm one of the organizers, that we really did not really have an ELSI theme within this meeting plan, but I do think with regard to the kind of amazing data that Amit and Laura showed, that one could easily, to get back to this issue of dissemination and communication, you can easily get into feeling like, well, I'm destined to be a smoker or to never be able to quit, or I'm destined to have obesity. So I do think it's very important how we communicate these kinds of results. And similarly, we talked in the first hour a lot about diversity, but it did not really come back up in this session. And I don't know how robust our data sets for these kinds of predictions are for obesity, for example, in other parts of the world, other populations, because I think that's an important piece. But I do think this issue of what are you destined to get versus predicting response to treatment are very different and need to be communicated pretty carefully.
Yeah, I think, Sharon, some of the data out there right now is that actually in terms of risk scores, if you don't derive them in a diverse population and apply them to a different sample with current methodology, they'll do very, very poorly. They'll give
Right, and I'm
predictions are
aware
way
of
out
that,
of
but
whack.
that didn't get raised in these presentations. So I guess my question to Amit is, that was amazing obesity data, but what population does it actually work in? And maybe we should make sure our presentations always make that clear.
Yeah, thanks for great points. The first has to do with disclosure of genetic risk information. And there I think that's something myself and many others are thinking a lot about, which is how would we design what might be a polygenic susceptibility report card for folks early in life? And what does it mean to be in the top decile of a score? At least for one example, again, I'm a cardiologist, so I thought a lot about heart attack. And we showed, at least in observational studies a couple years ago, that even if you're at high polygenic risk for heart attack, you can actually normalize that risk with adherence to a healthy lifestyle. And in unpublished data, we've seen the same trends with obesity and a few other diseases as well. But I think genetic risk disclosure is really ripe for innovation and patient engagement and genetic counselor engagement to really figure out how we're going to make it clear that people are at increased risk, but also make it clear that the DNA is not destiny. And I think there are a few use cases like heart attack or breast cancer with screening, or maybe a few other examples where we can make some early inroads. The second example, point you touch on is related to the application of genetic risk scores across various ancestries. And so I would say there are really two issues. One is that if you just look at the raw polygenic scores across different ancestries, the distributions are widely different versus things like blood pressure would be more or less the same. And there we've shown, at least for heart attack and a couple other diseases, you can essentially normalize the distribution just by taking into account genetic ancestry and principal components. So say, what would the genetic score be predicted to be based on your genetic ancestry, and how did you deviate from that, or what's the residual of that function? And that normalized the score remarkably well. The second issue is that what we observed is that the scores do predict risk in all diseases, but the gradient of risk is attenuated in the further you go, further away you go from European ancestry individuals. So there, I think. First, I would say that that's not a new problem in medicine. The Framingham Heart Study, which is the score we used for decades, was based on essentially Europeans in Framingham, and there was clear evidence that it didn't generalize across diseases. But so it's not new, but it's also not good. And so the two approaches, I think, one are computational approaches, where I think you can actually think about situations where you have even smaller data sets from diverse ancestry and potentially even incorporating local ancestry and take the weights accordingly. So that's the computational fix or hack to try to get at the problem. And the second is what's been highlighted already, which is ultimately we just need more data in more diverse populations. And so, I think the Million Veterans Program is a nice repository of African Americans, for example, and the Kadoorie Biobank in East Asian folks. And so I think we are seeing those results crop up, and I'm hopeful that a combination of computational and then expanded diversity will help address this problem. And I think in the meantime, I think it's just important to acknowledge that is a clear and present limitation in 2019.
So
David.
I want to make a comment about that too, because I think we have to recognize that private enterprise is going ahead with the polygenic risk scores. And they're going ahead very fast, and a company announced that it's going to do embryo selection based on polygenic risk score for educational attainment. So, the reality is this is coming. We should be doing the science, really leading that and what the issues are. And the issue about the concern of, I always call it the donut hypothesis, that, you're on the upper decile for obesity, you go eat the donut because, my genes are destiny. Or you're in the lower decile and you go eat the donut because, you're free. So either way, the donut is still bad. And
Yeah, but there are people that study the science of it. That's all I was going to say.
Yes, and
of
we-
whether people do one or the other.
Yes.
And we need to incorporate that.
And we need to change the paradigm because we don't have enough genetic counselors. And I personally am in the mode of changing the paradigm that it has to go through your healthcare professional.
All right, David.
I'd just like to sort of change the topic a little bit and come back to something that Jonathan mentioned, which was the challenges for education, particularly education of medical students and younger trainees. And I think we've lamented about this problem for quite some time. But I think actually right now we have tremendous opportunity. And I say that based on our experiences having medical students and fellows analyze real data that is relevant to their particular patients or to sample families that represent their patients. They can learn the bioinformatic tools. They learn the principles of genetics as they apply different models of inheritance. And we're just getting a lot more interest in genetics and genomics in our medical students now than we've ever had. And I think our discussions like we just had about genetic determination and ELSI issues also build into that. So I think we just need creative thinking about developing these educational tools, and I think that NHGRI certainly, for a small amount of money, could make a big difference there.
Thank you. All right.
So Dana Crawford, Case Western Reserve University. So I wanted to get back to the diversity issue, and so speaking directly about Laura's comments and some of the comments made about the generalizability of polygenic risk scores. So I think we all know in the community there are many limitations related to the lack of diversity in our GWAS samples. And I think it really takes us to point number 3 here is how are we going to get the samples needed for these studies to really make this truly precision medicine for all. So the Million Veteran Program, which was mentioned, is great. It's diverse, large sample size, but 95% of the samples are male or from men. So there's a limitation there. And so is the community waiting for the All of Us program to step in and fulfill that diversity promise? We haven't seen preliminary data on their recruitment and how well they're doing in that aspect. So again, where are we going to get the samples to really do the research that we need to get at this problem that we're having in diversity?
All right, so many hands. We haven't had any questions from that side of the room, so maybe Lynn, do you want to have your comment?
Yeah, thanks. Lynn Jordy, University of Utah. In the context of point number 3, sample and data collection, I'd actually like to go back to a point that Evan Eichler made earlier this morning about the difficulty in going from genotype to phenotype, understanding variation in expression. One of the contexts in which I think we can be more effective this way is in using family data, where people share varying amounts of DNA and where they share different levels of environmental risk factors. So I think it's important to emphasize still the value of families. I was pleased to see Jonathan's complex Amish family a few moments ago. But one area in which I think we've perhaps lagged a bit is in developing methods for efficiently and effectively analyzing family data, especially now that we have all sorts of large-scale omics data available potentially for families. So I think that's an area in which NHGRI could make a significant contribution.
Hi, Howard Ching from Stanford. This is a question for our panelists. Can you say a little bit about how somebody at your institution could actually make use of the genetic risk score? If I were a patient coming to an outpatient visit and I bring in my 23andMe profile, can you basically apply what you know, or what are sort of potential barriers to actually then making use of this information?
I'll tell you what would happen at my institution. The doctor would look at you and go, "Oh, crap," and really say there's nothing there that they could do, for a variety of reasons, because it's not FDA approved, it's not really in the medical record, there's no way to really interpret it.
Yeah, I would say that at the moment there are a number of issues that have been highlighted, but I think that one of the things that may not be the specific charge of this meeting is really to think about over the next 5 years, what are the most important clinical utility demonstrations that could be done? And one area that I'm particularly excited about is the use in thinking about screening technologies. And so, an example is a 40-year-old woman with average risk for breast cancer. It's actually quite controversial whether or not she should get a mammogram, because it actually does reduce breast cancer mortality, but the number of false positives is actually quite high. Similarly, in atrial fibrillation, there's actually been a renaissance in detecting these irregular heartbeats with new technologies like Apple Watches and others. And again, if you study a low-risk population, the number of false positives actually swamps out the signal. And then, even coronary plaque, which can be assessed via coronary artery calcification. Again, there are a number of technologies that actually have indeterminate utility in clinical medicine. And I think that using genetics, polygenic, monogenic, either to really enrich the yield of some of these screening modalities it could be quite useful. And then secondly, I think we ultimately have to show that if you disclose someone's genetic risk, that it actually impacts behavior in a meaningful way. I would say the studies to date have not suggested that people will go out and eat donuts or not eat donuts or so forth, and they can understand that this is one factor in a very complex disease process. But that really needs to be proven, and we need to show that in a rigorous way that we actually change people's behavior.
All right, Richard, you've been waiting for a bit.
Yeah, thank you, Richard Gibbs. In contrast to the previous session, it's harder here to see the distinction between what instantly falls under what we regard as the NHGRI portfolio and what's more general, solving all of the challenges of genetics and medicine. So maybe each of you can kind of reiterate what the sort of one thing that's really is NHGRI specific would not get done otherwise. That might be helpful. Or maybe it's not. Maybe it's a reflection on the state of the field.
I'll comment, which is I think we have to have some paradigm-changing approaches to animal studies. I think that they're too expensive now, and I think that that's general. I think we should be thinking of technology. I think back to the $1,000 genome. Have us have challenges about that. I think we need to think differently about how we return results. What are the paradigm changes kind of globally, not specifically? So I'm thinking about it in the sense of what are paradigm changes really that are general questions that cross all institutes and then, could be taken up and applied at the other institutes.
So, and I would just say, and this sort of harks back to something that Lynn just said, the computational approaches to handling all of these data. I mean, that is something that's across every institute, across every study, and that's something that we are clearly struggling with at every level. I mean, all the in silico annotation stuff, how do we deal with not just a single variant, but the whole haplotype or the whole gene region or locus region and those kinds of things. I think that's a fundamental approach that is something that we really need, that spans all of these different diseases.
You don't want to say? All right. All right. You've been waiting for a bit.
Yeah, I'd like to weigh in on Richard's question and also about the structural variation discussion that was happening. I think this is a real area where NHGRI could lead. I think I disagree with Gonzalo here, although I rarely do that. But I think, with regards to structural variation, the Centers for Common Disease Genomics has taken this problem very seriously. So we're trying very, very hard to do comprehensive trait mapping analyses, and it's very, very difficult. But I think there is a lot you can get out of short-read data for structural variation and repeat variation. But the problem is the tools aren't good enough, right? And so I think NHGRI needs to lead in building better tools for comprehensive trait association. And obviously, we'd rather be doing long-read sequencing. And so, we need an investment in technology to do better sequencing, to see all these things. And the third thing is that I do agree with Gonzalo that pretty much everything is imputable if we have solid enough knowledge of genome variation in populations. And that's the third way in which we could really make a difference, is doing an excellent job at documenting the variation that's out there so that we can actually impute it in much larger studies. And so we can't do all the trade association studies at NHGRI because there's not enough money, but I think we can lead in terms of actually showing how to do a comprehensive and thorough job of it.
I don't know where you disagreed with me, but if you disagreed on methods or technology, I want to agree with you on those points.
Well, you made a statement that most studies were either doing locus by locus or we're working with microarrays. And I would say that we're working in tens of thousands of genomes right now with whole genome sequencing data and doing trait associations with that. It's just that we're encountering technological challenges.
Okay, I think we should continue. But my point was different. It was more that when you do whole genome sequence data and then there's these examples of finding clear associations with disease, often it's not been from global methods that are characterizing variation in C4 and then associating it with schizophrenia. It's because someone took the whole genome data and spent time figuring out how to interpret it in the context of that locus. Anyway, Judy.
So I just wanted to comment in terms of where NHGRI can separate from the disease-specific institutes, and this is in response to a comment that Amit made, is saying that we have to prove benefit. And I'm actually saying that maybe this is where NHGRI can be at the forefront, because when I talk to people about the polygenic risk scores and I say, would you want to be told you're in the top, threefold increased risk for coronary artery disease, they say, yes, I would want to know. And so maybe the disease-specific institutes have to prove it, but I think, we should be at the forefront of assessing risk and communicating that to patients. It's a different paradigm of healthcare, but I think we should be at the forefront of that.
So can I just riff on that? So do we really have to show benefit? I think people want knowledge. They want knowledge even without benefit.
All right. Jay.
I'm a big fan of polygenic risk scores and the potential there, but I do, just to echo something Sharon said earlier, like the ELSI component in my mind is critical. I think the potential to do harm here, societally and otherwise, in many different facets, is almost equivalent to the potential to have benefit. And I think we need to think about this carefully. And this is, it's one place where NHGRI could take a leading role, in particular with the ELSI program. And I think where there is some urgency in particular, because this is moving so quickly in industry.
Thule.
Yeah, so I wanted to comment on the sort of something that is maybe a little bit missing somewhere there between phenotyping and genomic data, and that would be large-scale molecular phenotyping. When we can think about metabolome data that is now showing a lot of promise in understanding genetic associations to more complex phenotypes and disease, and then also large-scale transcriptomics data. That I think is in some ways being kind of sort of our success in projects like GTEx has been a little bit of a mixed blessing, in the sense that we've been very successful in mapping cis-regulatory associations, but then getting to the more complex trans associations that actually uncover gene regulatory networks and genetic effects on gene function in a cellular level. I think there is a lot of promise in investing in those kinds of initiatives in a very large scale. And we will be talking about tens of thousands of individuals that could be done in some of these cohorts, despite of some of the logistical challenges.
Yeah, I think that's a really exciting area, both for the observation, tens of thousands of individuals, and for figuring out how you do large-scale perturbations. The back there.
Eric Jarvis, Rockefeller University. So I have a question for Laura because you brought up the issue of privacy and how privacy rules might actually hinder scientific advance. And I'm wondering, though, could you give examples of what do you think needs to be done or fixed and how NHGRI can actually do that?
So I think I gave the example of like the FDA and kind of some of the regulations. So giving back gene results. So working with 23andMe, for instance, they were shut down by the FDA. But if they give results that are lifestyle, that's not medical. And so, one of the questions is, well, is smoking lifestyle? I think of smoking as actually a disease. So I'm also thinking about it is, how do we do all these genes at once, versus the type of testing that we've had before? Let me go on with privacy. The addiction field has been cut out of medicine in many ways, and we didn't have the ability to share any data based on addiction. It was outside of the medical record and required different types of testing. I could go on and on about the EHR. What I know is that the companies have a gazillion things of data, and we as investigators are often hindered in many things. And I really want lots of privacy, and I want everything, as private and shared as possible. But we have to realize that the industry is going ahead, really, really fast. And so it's really critical for us to think about this and to understand what's happening. And again, I see that with the Juul cigarette that's out there, is that we can't study it. I see it with marijuana. We can't study it. All these different things because of the rules we've set up.
All right. Gabor.
So I would like to bring up the concept of patient-specific reference genomes. Currently, genomic sequencing is done in the research domain to make discoveries, for example, gene discoveries. In the clinical domain, they treat it as a test. So we're doing sequencing to figure out what causes the patient's disease. But moving forward, obviously, everybody will be able to have their genome sequenced. So I'd like to explore the possibility of NHGRI investing in this concept moving forward. It's a cross-cutting proposal because we don't know necessarily what disease the patient will have in the future. It's longitudinal. It ties in with the polygenic risk scores. So having some sort of selection process and collecting genomic sequence, layering phenotypic data, functional data, as well as electronic health record data for a longer-term study.
Bing, do you have a comment?
I have a separate comment. Is it a good time to raise it?
Sure.
So I think we heard finding causal variant has been particularly hard, and I want to get the opinion from the panelists about what's the major bottlenecks that you see in that direction, and whether increasing molecular phenotyping in populations, as demonstrated by GTAX type of studies, would be a answer or a solution, one of solutions.
Does anyone want to tackle that?
Not really, but I think GTEx is a start. I mean, being able to look at the transcriptomics will help with those things, but it's only one way that a variant might have an effect on a phenotype. I think one of the big issues with going from loci to genes, if you will, is the naivete that a lot of people have about how to approach it. We still, and you saw it in my slide. I mean, I listed 51 loci. I listed it by the nearest gene, which is really a terrible thing to do, and I will admit it's a terrible thing to do. But how else do you put it on a slide to have people have any idea what you're talking about? So I think that there's, being able to look at, we need to be able to look at the whole context. When we have an association, what's the whole context of what's going on there? And some of that is getting more functional data in those regions. I mean, going back to, we need to be able to scale that kind of analysis so that we can look at it whole genome or at least whole regions very, very quickly and very easily. We need more reference data like GTEx and things like that to be able to look at that, because we may be able to use that information to move more quickly to it. I mean, there are a lot of issues there, and I think that spans like all diseases, so it's something that NHGRI I think really should be thinking about.
Do we have a recipe?
No.
I mean, there are recipes, but you need the ingredients are hard to get, so if you have,
all right,
you need both functional data, but you need good models for predicting function of individual variants, because often if they travel in blocks, the only way to distinguish it is with really good variant level predictions of function or incredibly large sample size, and probably Jay would tell you need large scale assays where you can modify each of those variants and get a readout. Eric.
So I'm having trouble synthesizing all this in terms of advice to NHGRI, but I'm going to try and then let you criticize me, or Eric can criticize you. One is on the translational side. I guess I'm hearing a clear message that the proverbial train has left the commercial station. And instead of labeling this as always an ELSI topic, it seems to me that NHGRI can work with other federal agencies to help shape policy a priori and help shape how this is going. My worry sometimes is the other agencies, the FDA is an example, but it's not the only example, left on their own may not shape policy given data and may not shape policy for the best interest of genomics and healthcare. So that's one side of it. The other side of it is, building off of Jay's talk this morning and then Tuli's question a bit ago, it seems like we could combine naturally occurring variation in humans like us and with molecular perturbations in mice and cells, and then very careful phenotyping. I think the word that was used is proximal, sort of, in people with known genotypes having good, sort of transcription data, proteomic data, levels and activity, and metabolomic data in quite large samples, and put that together. And I suppose the advice to NHGRI is to partner with something like the Common Fund to help get all that omics data in a large sample for elucidation of function to these genomic elements. And that, just hearing the chatter this morning and now, it seems like that's one way to put this together into a cohesive picture.
Thanks, Eric. Andrew, you've been waiting for a bit.
So I want to raise 2 points. The first one is that, and I want to go back to sort of the oh crap moment in 23andMe coming to a physician. I think much of the reason for that is because there are really no guidelines that are commonly accepted for actionability and clinical actionability of genetic information. So the question I think that we should ask ourselves, in cancer, for instance, we know that the NCCN has made out very clear criteria. If you have an area under the curve that is 0.7 or greater, then there is clinical actionability. People tend to try to prefer higher than that. But if you're below, it is not the case. So when I look at some of the polygenic associations that we're looking at right now, even with some of the most recent work Alex Young showed recently, for instance, that you can include even the genotype of the parents in doing those kind of polygenic associations, you still get to a predictability that is very low. So it is statistically significant, the odd-risk ratios are great, but in terms of clinical actionability, I think we're still very far away. And so this goes back to the second part of the question for the audience, is that I think that if we're looking at a space of 1,000 variants that contribute to a phenotype, where every possible combination of those variants can actually introduce different levels of risk, you're going into numbers that are greater than the number of atoms in the universe. And therefore, we're never going to be able to solve this problem statistically. We, I think, need to do this in a way that is more mechanistic. And I think Jonathan Pritchard, in human genetics, other groups in the context of cancer, proposed the idea that there are some proteins that actually act as integrator of the genetics, and not only of the genetics, but also of the environmental factor when we talk about nature of nurture. And so I was wondering whether actually a strong impetus should be given to studying things outside of the genome that are relevant for genome-mediated effects.
Okay. Shihong, do you want to make a comment?
I would like to make two comments, and the first is I think it's important to go from burden to function, but also it's important for us to think about how we should go from function to action, and in particular how we can collaborate with other institutions and also non-NIH institutions, and especially large healthcare systems, and incorporating the biobank data and then translate the knowledge into action, into the treatment target, and also into the prevention actions. And second is how can the NHGRI can help developing policy on reporting the result back to the participants, in particular the sequencing result. And right now there's no guidelines, and which is universally agreed on reporting the result back to the participants.
All right, I think we have time for one last question. So if someone is going to make a comment that's really new and different, maybe you can have a go.
Hi, Alvaro Montero from Moffitt Cancer Center. Just going to come back to the question from Ben Guren about the bottleneck. I think from moving from locus to gene, I think we've been very successful at using both eQTL and chromosome confirmation data. I think the bottleneck right now is that for a lot of the diseases, the cell type that is critical to the disease is actually a minority cell type in a tissue. So I think we definitely need to move both eQTL and confirmation capture to single cell so that we can kind of really identify things that are specific. Case in point, for example, melanocytes in skin.
All right, I'll leave you the last comment.
Yeah, so if somebody is taking a list of the action items to do or to not do, one of them is probably the next big push is going to be the SpaceX equivalent for the genome. It's going to come out of private hands. Are there any roadblocks that can be removed by these efforts, the Common Fund and NHGRI, for access by AI tools such as Watson or others that are being developed, and that we want this rocket fuel, if you will, being made in this room to be part of those efforts? So it's removal of barriers.
Thank you.
Something to think about over lunch. We do need to break now for lunch. I want to first thank the speakers and the moderator and all of you for a great discussion. And I have an announcement about lunch. If you've paid for a boxed lunch option, the lunches are available outside this room, and they're individually labeled and grouped by the first letter of your last name. And if you did not pre-order lunch and need assistance finding food options, please ask the meeting staff at the desk outside. And we're going to start promptly at 2 p.m. Thank you.
So we have an hour for lunch.
We have an hour for lunch.
Ah, good.